This PowerPoint will look at DNA replication in detail. DNA replication depends on an enzyme called DNA polymerase. This is the enzyme that synthesizes the bonding between nucleotides, making the backbone of the DNA molecule. This is the enzyme that synthesizes the sugar phosphate sugar backbone of the DNA molecule. However, DNA polymerase has two constraints. One, it can only replicate DNA proceeding in the 5' to 3' direction. Second, DNA polymerase cannot start DNA replication. It can only continue adding nucleotides from an existing strain. Let's look at the first constraint. DNA polymerase has to proceed in the 5' to 3' direction. If this is the parental strand, to copy this strand, the DNA polymerase will have to move in the 5' to 3' direction. The reason why replication has to proceed in that direction is because DNA polymerase has to add the new nucleotide to the 3' carbon of the existing nucleotide. So the phosphate of the new nucleotide is covalently bonded to the 3' carbon of the existing nucleotide. The nucleotides will always be positioned 5' to 3' in order for the phosphate to have access to the 3' carbon. Therefore, the newly replicating chain will be replicating in the 5' to 3' direction. Here we have another nucleotide added to the chain. Replication proceeds in the 5' to 3' direction. As the nucleotides are positioned 5' to 3', making the 3' carbon of the existing strand accessible to the phosphate group of the new nucleotide. This is the new nucleotide. This is the already existing nucleotide strain that is being replicated. The new nucleotide has to be in the 5' to 3' direction for the phosphate group to be covalently bonded to the 3' carbon of the already added nucleotide. The next nucleotide will be added right here, and the chain will continue to grow in this direction, 5' to 3'. To further make this point, notice what would happen if we try to add nucleotides in the opposite direction, that is 3' to 5'. That means that the newest nucleotide would have to be added to the 3' carbon of the existing nucleotide. The bond would have to go from the phosphate across this phosphate to this 3' carbon in order for the chain to continue to grow in the opposite 3' to 5' direction. Notice that the two molecules that have to bond are too far away from each other for that to occur. Therefore, DNA replication cannot proceed from 3' to 5' direction. It has to proceed 5' to 3'. The initial process of DNA replication Notice that in DNA replication, the parent strands are going to serve as templates from where the complementary new strands will be synthesized. DNA replication proceeds bidirectionally. Remember that replication begins at a specific point in the DNA. This point is called the origin of replication. From the origin of replication, DNA is replicated proceeding on both directions of the origin of replication, with each parent strand serving as templates. To the left of the origin of replication, there will be a 5' to 3' replication using the 3' to 5' parental strand as the template. The 5' to 3' strand will serve as the template for a replication proceeding in the opposite direction. To the right of the strand of the origin of replication, the same is happening. DNA cannot start synthesis, it can only continue an already started strand. This is the second constraint of DNA polymerase. For this reason, another enzyme called a primase needs to first synthesize a short nucleotide sequence called a primer. From the primer, DNA polymerase can continue synthesis of DNA. This blue line represents the primer. The primase synthesizes the short sequence from which DNA polymerase can continue elongating and synthesizing DNA. Notice that each strand is going to need at least one primer. Because DNA strands run antiparallel, the new strands are synthesized differently. The new strands being synthesized away from the origin of replication, like this one and this one down here, can proceed uninterrupted and are synthesized continuously. These strands are called the leading strands. Notice that the leading strands synthesize DNA using the 3' to 5' parent template. 3' to 5' is the template that allows for synthesis of the 5' to 3' continuous strand or leading strand. DNA strands that are synthesized towards the origin of replication have to be synthesized in fragments called Okazaki fragments. These strands are called the lagging strands. Take a look at down here, this strand right here. This first fragment has a primer. From the primer, DNA polymerase begins synthesizing DNA, and as it continues synthesizing DNA, it will eventually run against the previously synthesized strand right here. Therefore, DNA polymerase has to stop, jump backwards. Primase will synthesize a primer. DNA polymerase will begin to synthesize the DNA, and it continues on until it runs against the previously synthesized fragment. DNA polymerase has to again jump backwards. Primase will synthesize a primer. DNA polymerase will begin synthesizing from the primer until it runs against the previous fragment. Notice that this strand is synthesized in fragments called Okazaki fragments. This is the lagging strand. The lagging strand uses the 5' to 3' parent template as its template to synthesize its DNA. 5' to 3' parent template is being synthesized in fragments. Primase, DNA polymerase begins synthesizing from primates, and it will continue until it runs against the previously synthesized strand. Then it has to jump backwards and start yet again. Another primer, and DNA polymerase begins synthesizing from that primer and continues until it runs against the previously synthesized strand. And thus it proceeds, creating these Okazaki fragments. The initial process of DNA replication occurs wherever DNA is located in prokaryotes and eukaryotes. It begins at the origin of replication, which is 1 in the case of prokaryotes and many ORs in the case of eukaryotes. From the origin of replication, a replication fork is set up with all the enzymes needed for replication. First, histones have to be removed and the DNA have to be unwound, so the histone proteins will be removed. Unwinding of DNA takes place by enzymes called DNA helicases. Here's 2 DNA helicases unwinding DNA. The 2 DNA strands will be kept apart by stabilizing proteins. Here are the stabilizing proteins keeping the 2 DNA strands apart. A DNA polymerase enzyme binds to each strand on either side of the origin of replication. We're looking at only one side of the origin of replication. Therefore, we have 2 DNA polymerase, 1 for each strand. Remember, we will need a primase to synthesize the primer at the beginning of each strand. DNA polymerase can continue synthesis of new strands by adding nucleotides from the primer on forward. Remember, the 3' to 5' DNA parent strand serves as the template to the leading strand, which replicates continuously. This is the DNA polymerase replicating from the origin of replication somewhere in this area continuously. Here's the primer and there's the already replicated strand, which continues to be replicated by DNA polymerase. Synthesis of the lagging strand will continue on in fragments as it moves towards the origin of replication, which, as we said, was somewhere over on this side right here. So the 5' to 3' DNA parental strand serves as the template to the lagging strand, which replicates discontinuously. A primase synthesizes a primer for each Okazaki fragment, and DNA polymerase extends from the primer, proceeding as always in the 5' to 3' direction. DNA polymerase has a unique quality. It has proofreadability. Proofreadability means that it can check for mistakes as it's replicating DNA. For example, if it introduces the incorrect nucleotide, it can check and replace the incorrect nucleotide for the correct nucleotide. This means DNA polymerase seldom makes mistakes. It only makes a mistake 1 in every billion nucleotides it adds to a DNA strand. You should now be able to answer the following questions. Make a list of enzymes needed for DNA replication and their function. List the characteristics of DNA polymerase enzyme. Given the double-stranded DNA sequence below, determine which strand is the template for the leading strand and which strand is the template for the lagging strand. The origin of replication is to the left of the strand. Direction of replication proceeds in the direction shown by the arrow.